The overall goal of this procedure is to accurately image endosomes in three-dimensional space over time, or in 4 dimensions. This is accomplished by first dissecting snake muscles and arranging the tissue in the imaging dish. The second step is to stain the tissue with vital dyes to stimulate endosome formation. Next, the preparation is imaged in 4 dimensions for 1 or more neuromuscular junctions. The final step is careful data analysis to characterize the behavior of macroendosomes. Ultimately, the presence and movement of endosomes over time within the presynaptic space can be demonstrated by 4-dimensional fluorescence imaging.
The main advantage of this technique is that the snake abdominal muscle is only a single fiber thick, which allows for high-quality images to be generated using standard epifluorescence microscopy.
Tissue preparations from the garter snake will be used in this protocol. Reptilian tissue should be kept at low temperatures so it remains physiological for longer times, and with less bacterial contamination. For lysosomal vital dye staining, the dye is dissolved 1 to 5,000 in cold reptilian Ringer's solution to get a final concentration of 0.2 micromolar. Add the dye to the tissue and incubate at 4 degrees Celsius for 15 minutes. After 15 minutes, wash the tissue several times with cold Ringers. For all FM143 staining, pre-mount the sample in an imaging dish with magnetic pins prior to staining. For the purpose of this video, only FM